Now diving deeper, the diagram shows a monohybrid cross in mice and explains step by step how traits are passed from parents to offspring based on Mendelian genetics. First, in the parental generation, a homozygous black male is crossed with a homozygous brown female. The male has 2 dominant alleles for black fur, while the female has 2 recessive alleles for brown fur. Next is gamete formation during meiosis. In the male, cells in the testes divide to produce sperm cells. Since the male has 2 dominant alleles, every sperm cell carries 1 dominant allele. In the female, cells in the ovaries divide to produce egg cells. Since she has 2 recessive alleles, every egg cell carries 1 recessive allele. During fertilization, a sperm cell from the male combines with an egg cell from the female to form a zygote. Each offspring receives 1 dominant allele and 1 recessive allele, making all of them heterozygous. In the first generation, all offspring appear black because the dominant allele masks the effect of the recessive allele. Therefore, all offspring in this generation are heterozygous and show the black phenotype. Now before we continue, it is important to understand the terms P generation and F1 generation. The P generation stands for the parental generation. These are the original parents used in a genetic cross. In many basic examples, both parents are homozygous, meaning they have 2 identical alleles. For example, one parent may be homozygous dominant while the other is homozygous recessive. When these 2 are crossed, each parent contributes only 1 type of allele, so all the offspring receive 1 allele from each parent. As a result, the offspring are 100% heterozygous. The F1 generation stands for the first filial generation. These are the offspring produced from the parental cross. Since all offspring are heterozygous in this case, they all have the same genotype and show the dominant phenotype. This generation helps us understand how traits are expressed when dominant and recessive alleles are combined. Next, we will see what happens when the offspring of the first generation are bred together, as this diagram shows the next stage of the process. The diagram represents a monohybrid cross between 2 mice that are heterozygous for coat color. It shows how traits are passed from these parents to their offspring. In this case, black coat color is the dominant trait, while brown coat color is the recessive trait. Both parents are heterozygous, meaning they appear black but each carries 1 recessive allele for brown. During meiosis, each parent produces 2 types of gametes. Half of the gametes carry the dominant allele, and the other half carry the recessive allele. When fertilization takes place, these gametes combine randomly, producing 4 possible genetic combinations. One combination is homozygous dominant, which results in black fur. 2 combinations are heterozygous, which also result in black fur because the dominant allele is expressed. One combination is homozygous recessive, which results in brown fur. As a result, the phenotypic ratio is 3 black to 1 brown. This means there is a 75% chance that an offspring will have black fur and a 25% chance that it will have brown fur. The diagram shows 2 different genetic crosses involving mouse fur color, where capital B represents the dominant allele for black fur and small b represents the recessive allele for brown fur. In the first cross, a homozygous black mouse represented by capital BB is crossed with a homozygous brown mouse represented by small bb. The black parent has 2 dominant alleles and can only produce gametes carrying the dominant allele, while the brown parent has 2 recessive alleles and can only produce gametes carrying the recessive allele. When these combine, all offspring receive 1 dominant allele which is capital B and 1 recessive allele which is small b, making them all heterozygous. Because the dominant allele is expressed, all offspring appear black. So, the result is 100% black offspring with the same heterozygous genotype. So, from this we understand that when 2 homozygous individuals are crossed, all the offspring produced are heterozygous, resulting in all black and 100% heterozygous offspring. In contrast, the heterozygous parent produces 2 types of gametes, one carrying the dominant allele and one carrying the recessive allele, while the brown parent produces only recessive gametes. When these combine, half of the offspring become heterozygous and show black fur, while the other half become homozygous recessive and show brown fur. This gives a 1-to-1 ratio, meaning 50% black and 50% brown. In summary, when a homozygous dominant is crossed with a homozygous recessive, all offspring are heterozygous and show the dominant trait. But when a heterozygous is crossed with a homozygous recessive, the offspring are divided equally between dominant and recessive traits, giving a 1-to-1 ratio. So, the F1 generation refers to the first group of offspring produced from the parental generation. In this case, all individuals are heterozygous, meaning each one carries 1 dominant allele and 1 recessive allele. These F1 individuals then act as the parents for the next cross. The F2 generation is the second group of offspring produced when the F1 individuals are crossed with each other, meaning heterozygous crossed with heterozygous. In this generation, a 3-to-1 ratio is observed, where 3 offspring show the dominant trait and 1 offspring shows the recessive trait. This occurs because fertilization is random, and each possible allele combination has a certain probability of occurring. A large number of offspring is needed to clearly see this 3-to-1 ratio, because small samples may not always reflect the expected genetic pattern accurately. Think of generations like a family line. The P generation, or parental generation, are the original parents used in a genetic cross. For example, a homozygous black parent is crossed with a homozygous brown parent. The F1 generation, or first filial generation, are the offspring produced from the P generation. From a cross like capital BB crossed with small bb, all the F1 offspring are capital B and small b. These are the first children of the cross, and they are all heterozygous and show the dominant trait, which is black in this example. F1 individuals are usually not shown inside a Punnett square because they are the result of the first cross rather than part of the prediction process. So, when a homozygous black male is crossed with a homozygous brown female, all the F1 offspring are capital B and small b, meaning all appear black because the dominant allele is expressed. When these F1 individuals are later crossed with each other, the probable ratio of coat color in the next generation becomes 3 black to 1 brown. So the simple rule to remember is, the F1 generation is the first group of offspring produced from the original parental cross. For example, when a homozygous black male is crossed with a homozygous brown female, all the F1 offspring are heterozygous and appear black. The F2 generation is produced when the F1 individuals are crossed with each other, meaning the F1 generation essentially produces the F2 generation through self-crossing or interbreeding. To picture it easily, the parental generation produces the F1 generation, and then the F1 generation crosses within itself to produce the F2 generation. This means the F1 parents are responsible for producing the F2 offspring. In the F2 generation, the probable ratio of coat color is 3 black to 1 brown. The Punnett square shows the possible combinations of capital B and small b alleles contributed by both parents. Both the rows and columns represent capital B and small b, showing all possible pairings between the alleles. The results inside the square are capital BB, capital B and small b, capital B and small b, and small bb. This shows that both parents have the same genotype, capital B and small b, meaning they are heterozygous, so the genetic cross is written as capital B and small b crossed with capital B and small b. In terms of generations, the parents involved in this cross belong to the F1 generation. Their offspring, which have the genotypes capital BB, capital B and small b, capital B and small b, and small bb, belong to the F2 generation.